Hi class. Today we're going to dive into my favorite subject in bioinformatics, and that is the analysis of protein structure. Now, with protein sequence analysis, we were able to look at the basics of protein sequences in terms of the biology. And we then applied certain bioinformatics concepts to the study of protein sequence. So I want to do something similar with protein structure. And the reason is that the protein data bank, which is the repository for protein structure, is actually quite complicated unless you know a few basics about protein structure. Now, I know most of you have probably had formal education in biology and biochemistry of proteins, but I thought it was useful for us today to spend some time understanding and remembering some of our protein structure biology. And this will lead us nicely into the next lecture, where I can discuss more of the computational elements of studying the protein data bank. So today I want to look at the 4 levels of protein structure initially, and then we're going to look at each level in more detail. So primary structure, so the one with the degree symbol, remember, is primary. Secondary structure, tertiary and quaternary structure, and finally we're going to just very briefly look at the protein data bank, which will leave us in a nice place to continue on in the next lecture. Okay, so I've already alluded to the fact that there are 4 levels of protein structure. And this is a nice overview slide. By the way, there are going to be a lot of figures today, so I hope that is somewhat stimulating and engaging for you. Just in case you know this material, looking at the pictures will very quickly remind you of this material when you're studying for this later. So here's our first big image. Primary structure is the chain of amino acids. So there are some amino acids connected together. They then adopt some very particular structural motifs. In this case, this is known as an alpha helix. The other main structure that they can adopt is something called a pleated sheet or a beta sheet. And that sort of looks like this in terms of the bonding. Okay? This second level of structure is then further coiled and folded upon itself to form tertiary structure, which is the 3-dimensional structure of the protein. And quaternary structure is just a combination of multiple tertiary structure subunits. So in this case, we're looking at hemoglobin here. Just as a side note, the heme units, bonus points if you can tell me what metal is found in a heme unit. Think of what hemoglobin does in the body. Okay, let's take a look at primary structure. Primary structure is composed of amino acids that are connected together by peptide bonds. And each amino acid is defined by its side chain or R group. So I will use the term side chain, R group, side group. These terms all mean the same thing when it comes to amino acids. So here's our second picture. You might be asking yourself, what exactly is a peptide bond? So here are 2 amino acids, okay? We don't know what they are, but we do know that they have some chemical property in their R groups. So to simplify the figure, we use the term R to denote the side group. And an amino acid has a very particular topology. It has what's called an amino group on this side, the NH2, and it has a carboxy group or a carboxyl group, which is a CO, double bond O, single bond to the OH. And the carboxyl or the carboxy group of one amino acid binds to the amino terminus of the other amino acid, and the reaction results in the release of OH and H in the form of water. So this is a dehydration reaction. So dehydration, remember from your chemistry, means the release of water. So when you feel dehydrated, you're lacking water. So in this case, we are releasing water to form this peptide bond. And what you get here is a peptide. So this is the first amino acid, and this bit here, I'm not sure if you can see this light yellow, but this bit here where the bond formed, this is known as the peptide group. Okay? Oh, and this particular bond here between the C and the N is known as the peptide bond. So amino acids are connected to each other with these peptide bonds. In each case, one water molecule is released, and this is what the primary structure is. Furthermore, in a peptide, one end of the peptide will still have an exposed amino terminus of the first amino acid, and the other end will have the exposed carboxy terminus of the last amino acid. So in a single amino acid, you have a certain polarity with N on one side and C on the other. But in a peptide or a group of amino acids, you still have the amino terminus, or what's known as the N terminus because of the nitrogen. And you have the carboxy terminus, or the C terminus. So proteins have a direction, if you will, where the N terminus then leads to the C terminus. So generally, when you speak about the primary structure of amino acids, you normally refer to it from the N to the C end. Okay? So as I mentioned, we have these R groups, or side chains. There are 20 standard, naturally occurring common amino acids. There are more modified amino acids. There are 21 and 22 and 23 amino acids. There's one very particular kind of amino acid which is quite rare, which is known as selenocysteine. And this is considered to be the 21st amino acid. You might have also heard of something called taurine, which is found in your energy drinks like Red Bull. That is also an amino acid, and it's a modified amino acid. But for all intents and purposes, when we're looking at protein structure, we are dealing most commonly with the 20 amino acids shown here. And all of them have a common N and a C and a hydrogen. All of them have an N, C, and a hydrogen. The only thing that's different is this R group down here. Okay? Glycine is unique in that its R group is just a hydrogen. Okay? But all of the other ones have something more than just a hydrogen. And some of these are very intricate structures, and they're quite large. Furthermore, the side groups also have polarity, or they can be nonpolar, or they can have an actual physical electrical charge, where you have acidic, where you have negative charges on the side groups, and you have basic, where you have a positive charge, usually because of a nitrogen, and in the negative case, you have a carboxy group down here. So again, don't worry too much about the chemistry of this. Just know that there are nonpolar, polar, and electrically charged amino acids that are all created as a result of their different R groups. The other thing to remember when it comes to folding for proteins is that when you have large amino acids like tryptophan and phenylalanine, they can affect folding in certain ways just because of physically the amino acids take up more space, and so it does not promote folding in some directions, whereas in other directions, folding is much more favorable. So we'll come to that when we discuss higher level structure, but it's something to be aware of now because we're talking about the R groups. Just a note here, proline is very interesting because the R group is actually bound to the N terminus. Okay, so that's the only one which does that, and it's interesting. Proline has some very interesting properties when it comes to protein structure. So the primary structure is denoted by something you've already seen before, which is the amino acid sequence. So the sequences that we got from UniProt can be thought of as describing the primary structure of those proteins. So then what is secondary structure? Secondary structure is formed by hydrogen bonding between non-adjacent amino acids in a peptide. So adjacent amino acids are connected together by a blank bond, a peptide bond. Secondary structure, you have non-adjacent amino acids that form hydrogen bonds with each other. And repeating patterns of hydrogen bonding forms well-known secondary structure. And the two most common cases that you will encounter are helices and sheets. So here we have another image. As I said, we have a lot of pictures today. This figure was taken from a Nature educational section of their website, which I highly recommend, actually, if you want to brush up on various topics. But in this case, here is an amino acid. It has the amino group and the carboxyl group, and the side chain is the R over here. Okay, and here it is shown in a peptide. So here's the peptide bond. This is the next peptide bond. This is the next peptide bond. So we have 4 amino acids here denoted by 4 different R groups. Okay, and they form part of the structure. So part of this peptide is formed of these 4 amino acids. And what that does is you start getting hydrogen bonding between non-adjacent amino acids. So remember, these are the peptide bonds, and here are the dashed lines here are the hydrogen bonds between non-adjacent amino acids. And you start getting some kind of a pattern that is not just random, it is more rigid, it is held together by this hydrogen bonding. Okay? So let's take a look at alpha helices and beta sheets, because those are the most common structures you're going to encounter. There we go, another picture for you. What we have here is an alpha helix where you're seeing the amino acids wrapping around in this helical structure. And you are seeing in yellow the hydrogen bonding that is causing the structure to remain relatively stable. And in a beta sheet, it's known as a pleated sheet because it does actually form this pleated kind of ridged, wavy structure. And that's just because amino acids, as I mentioned, R groups do take up space. And so for these bonds to form, the amino acids do have to arrange themselves in slight pleats. So they're not in a flat line. They do have some three-dimensionality to them. So here again, we're seeing the hydrogen bonding between a hydrogen of this amino acid here and the oxygen of this amino acid here. Okay? And this picture was taken from a website called studyblue.com. Alpha helices are extremely common, and they're denoted by a bond between the ith and the I plus 4th residue. So the hydrogen bonding will happen between, let's say this is I, then if you follow this helix around, this will be the I plus 4th residue. And beta sheets are also quite common, and the bonds repeat regularly. Okay, so I don't want to get into it too much more, but there are other helical and beta types of structure. Okay, so the alpha helix is one kind of helix. The alpha means it's the I and the I plus 4th bond. You have pi helices. You have what's called 310 helices, which are extremely rare. And you also have left-handed alpha helices, where the actual rotating pattern, you have left-handed helices where the rotating pattern itself is going in the other direction. So those are extremely rare. And beta structure is actually very, very complicated and very diverse. So you can have sheets. We've seen one kind of beta sheet, but you have 2 kinds of sheets. You have parallel and anti-parallel. You have beta sandwiches, beta propellers, beta barrels, and you also have beta helices. And here's an example of a beta helix, where the arrows denote beta structure, and it actually does form a superstructure, which is a helix. So this is extremely rare, and furthermore, it's a left-handed helix. which doesn't really happen in nature. So one property of amino acids is that they are right-handed in nature. So life is right-handed. But this, occasionally you do get left-handed motifs, and they are formed of right-handed helices and right-handed amino acids, pardon me, but the way they're arranged causes them to coil around in a left-handed manner. Okay? And this is from a structure 1QRE, and this is the PDB entry for this structure, and we'll come to that a little bit later. So this is a beta helix, which is extremely rare. In fact, this is the first time I even have heard of one. When I was researching to put together this lecture, I realized that beta structure can also include helices. Okay, I'm just going to take a sip of water very quickly. Right. Tertiary and quaternary structure. Now, tertiary structure is what you're used to seeing when you see a 3D model of a protein. And they're formed through hydrophobic interactions between amino acids. So it's not between adjacent amino acids. There isn't necessarily a repeating pattern. It is literally just amino acids folding in. This is the spontaneous folding where hydrophobic amino acids tend to come together. So remember what hydrophobic means. Hydrophobic means hydrophobia, which is the fear of water. So these are amino acids that are, in terms of their charge, are they polar, are they charged, or are they non-polar? Going to give you a second. So these are interactions between non-polar amino acids. So non-polar amino acids will generally be found inside the structure of a protein, inside the three-dimensional structure. And they interact with each other, and these are called hydrophobic interactions. They're also affected by charge and polarity on amino acid side chains. So in the case of hydrophobic interactions, you're going to have amino acids without any charge burying into the structure of the protein so they can be far away from the water. But on the other side, you have charged amino acids that like water that are going to be found more likely on the surface of the protein. Now again, these are general guidelines. There are many cases where you have nonpolar amino acids on the surface. There are some very specific cases where you will find nonpolar amino acids in membranes, in membrane proteins, where they essentially help stabilize membrane proteins, because think of cell membranes as being very different than simply cell cytosol. So cytosol is essentially effectively mostly water. So proteins that are found in this aqueous environment will tend to follow these rules. But remember, there are exceptions for everything. And not all folding is spontaneous. So we think of proteins folding spontaneously into their 3D structure when an amino acid peptide chain is thrown into some water. You expect it to just fold up into its structure. But there are actually chaperone proteins inside the cell that help stabilize different portions of the amino acid chain in order for the folding to happen correctly. And finally, when you have multiple tertiary subunits, so each tertiary blob or 3D structure is known as a subunit, they can interact in similar fashion through hydrophobic interactions. In some cases, there are actual bonds between cysteine residues. Cysteine is an amino acid which has sulfur in it. So if two cysteines come close together and the environment is correct, the two sulfurs can have a bond, and this is called a disulfide bond. So in some cases, quaternary structure is stabilized through disulfide bonds, but mostly it is through hydrophobic interaction. So quaternary structure is the combination of multiple subunits of tertiary structure. Now, it doesn't mean that every subunit needs to be the same. In the case of hemoglobin, there are 4 subunits that form the full structure of hemoglobin. 2 of them are called the alpha subunits, which are identical, and the other 2 are called the beta subunits. So each subunit doesn't have to be like all the other subunits. The point is that quaternary structure is formed in this modular fashion through multiple interactions between various tertiary subunits. Also, hemoglobin being a good example of this as well, quaternary structure could also require metal ions or other cofactors to stabilize the structure. So remember that picture of hemoglobin that we saw? I had asked you a question. What is the metal ion inside hemoglobin? Well, what color is your blood? It's red. Hemoglobin is in your blood. What does hemoglobin do? It carries oxygen. Oxygen is a negatively charged atom, or negatively charged ion, in fact. And so hemoglobin, in order to carry oxygen, needs to have a strong positive charge that the oxygen will be attracted to in order for hemoglobin to do its job. And the metal ion that gives the color red to your blood is iron. So there is a small iron ion sitting in the middle of each tertiary subunit of hemoglobin. So one hemoglobin molecule, one hemoglobin protein structure, which is formed of 4 of these tertiary subunits, can carry 4 atoms of oxygen. And it's really at this level of the tertiary and the quaternary structure where you start seeing protein function. So this is the case where protein structure gives rise to its function. So, in cases where you have strong acids and strong bases, you might have heard the term denaturation, which means that the proteins tend to unfold in these strongly acidic and basic environments. And the way that these studies are usually conducted is that they try to see if the protein maintains its function under these different conditions. So by knowing that a protein loses its function, one can assume that the protein has lost its folding. In other words, on the flip side, protein structure determines function. Okay? And this just illustrates the point I just said, where misfolding or denaturation reduces or removes protein function. So let's visualize what this looks like. You might have seen very pretty pictures of protein structure, and they're all formed through different visualization packages, software. Helices are usually represented as springs, and sheets are represented as arrows, as we saw in the beta helix example. And the arrows show the directionality of the sheet, in other words, which way the bonding is happening. But there are other ways to visualize. So here is an example of a helix that's rendered with the program called PyMol. Pi should give you a clue that it's written in Python. So it's actually quite a useful package to interact with BioPython, for example. And what we have here is an alpha helix where you're seeing the bonding in yellow. And this is a backbone and a ball model. So the backbone is this gray kind of springy structure. And the ball model is showing the interactions between the atoms. Okay? So different visualizations do exist, but the most common one tends to be the coiled or the ribbon diagram, where helices are represented as fat ribbon springs and sheets are represented as thick arrows. Okay, why did I go through that exercise? It's all to lead to this, the protein data bank. The protein data bank is the de facto, or the comprehensive, the general repository for protein structure. What's very interesting about it is that it's actually very, very old. It's older than the field of bioinformatics. Think about that for a second. You've got a computer database that was created in the 1970s, and it is older than the field of bioinformatics. And even back then, they knew that it was important to have a file format standard for storing protein structure. And it is still that standard, for the most part, mostly unchanged, that we use today to describe protein structure. It's incredible. And as with all good databases, it is mirrored in 3 different places. So we have RCSB in America, PDB-E for Europe in Cambridge, and PDB-J for Japan in Japan. And they all fall under this umbrella, which is the Worldwide Protein Data Bank, WWPDB. Bonus points for anyone who knows what the PDB used to be called back in 1970. I'll let you think about that and get back to me next week if you have an answer. And the PDB is responsible for the PDB file format. And the PDB file format contains almost anything you'd want to know about the structure of a protein. It stores sequence, structure, and experimental information in different sections. So this is the data. And what else does it store? What is always accompanying data? Think about that for a second. The answer is metadata. So remember that data by itself is actually not very useful. We need to have the metadata to understand what it is exactly that we are working with. And each PDB entry is characterized by a 4-character PDB ID. And this is the best way for you to get access to a structure. Every publication, every paper, every poster that talks about a protein structure will contain a PDB ID if the structure has been published. Okay? So I'm going to give you a brief snippet of what the PDB file looks like. And in particular, I'm going to look at a section of the PDB that's going to be very relevant to us next week. And these are called the atom records. So this is taken out of a PDB file, okay? Atom records are the section of a PDB entry that store actual atomic coordinates for amino acids inside a protein crystal. Now, we're going to come to the actual methods of how do you actually get the structure of a protein next week. But suffice to say that one main method of determining protein structure is through crystallization. And in a crystal, you've got different copies you've got hundreds of thousands of copies of the same protein that's been crystallized. And because it's been crystallized, the positions of the different atoms are fixed. So given a frame of reference, we can say that this hydrogen is located at coordinate XYZ. This carbon is located at X plus 1, Y plus 1, Z plus 1. So these are actual atomic coordinates for amino acids inside a protein crystal. And this is how visualization packages like PyMol know how to draw the 3D structure. It's through the atom records. So let's take a look at what these look like in a file. So each amino acid is broken down into each of its constituent atoms. So remember, this is an atom record. We're not looking at the full amino acid here. We're looking at individual atoms. So here is the N for aspartic acid, and here is the carbon alpha, then the second carbon and the oxygen, and aspartic acid has a second carbon and a third carbon called the C beta and C gamma. And then there are some extra oxygens in aspartic acid as well. And each line is describing the 3D coordinates for each atom. Think about an atom, okay? We're talking about each atom here. We're not talking about the entire amino acid in one go. And And this is what the basic atomic coordinates looks like inside the PDB format. So each line is a single atom record. So the set of them are called the atom records in plural, okay? So if you talk to a crystallographer, please make sure you use the plural for atom records. This is a mistake I used to make a long time ago, quite frequently. The entire set of records is known as the atom records, okay? And the file structure, if you notice, is very, very fixed. So there is a fixed set of spaces between each of these elements here. And in fact, the PDB format is very, very restrictive in how many columns of data it has and how wide the file actually is. And the reason this is important is because back in the day when computers didn't have as much memory, one of the ways you reduced the amount of memory you needed, or you didn't know you needed, was knowing exactly how wide a line was that you were going to read. That way, programs could be written very efficiently to directly pick out the exact portions of a line where you knew this information was going to reside. Now, we'll come to this more in more detail next week. So this is my last slide in the lecture today, but I thought it would be helpful for us just before we broke today to look at the protein data bank, just briefly. So this is the WWPDB, and this is the umbrella project for the Worldwide Protein Data Bank. And it acts as a centralized location, but generally you'd be accessing one of these mirrors. Now actually there is a fourth mirror that's just been launched recently, but this is actually for a subset of the PDB, so we're not going to talk about the BMRB today. So for us, we do have the RCSB, the PDB-E, and the PDB-J. Now I reckon for you, because these are mirrored, the RCSB is probably going to be the closest to you and will probably run the fastest. And because they're maintained by different projects, you'll see that this is the front page for the RCSB, and this is the front page for the PDB-E. You notice they're very different, but believe it or not, at the end of the day, they all rely on these PDB files that are deposited and that are updated every single week. So everything you see here is basically metadata that's designed to look nice and make it easy for you to interact with the information on this database. So next week, we're going to look more at using these web pages, but we're also going to look at the actual PDB files and maybe a little Python example, if time permits, for how we can extract some useful information from a PDB file. By the way, the Ebola virus is now very, very big news at this point, and there are many groups that have been depositing Ebola virus structures. So if you want to look at what an Ebola virus, portion of an Ebola virus or a protein associated with an Ebola virus looks like, go to the PDB and just click around and start playing with it. Now again, I'm hesitating going into great detail here, but basically you can see that there's a section where you search for things, and that this will give you more data on the actual structure of the protein. And again, we'll look at this in more detail next week. Okay. Before we break off, I just want to recap a little bit about protein sequences. Now, I haven't received any questions from you, and there is an in-course test that's happening soon. So now would be a good time for you to email me with any questions you have up to this point about any of the things that we've discussed, because I'm more than happy to respond to you, and we could even set up a Skype chat or something like that if you'd find it useful. Or send your questions through Dr. Jaya or through your classroom representative. Again, I'm looking forward to hearing from you, because your feedback is always very helpful to me. Right, so without further ado, I will let you go, and I hope you enjoyed this lecture. It was very different, and again, remember, we had to focus on the biology today in order to understand the bioinformatics for next time. So I hope for some of you this was a nice, refreshing review of protein structure. But I hope some of you got some new tidbits out of it and some new ways of thinking about protein structure. Remember, I try to present everything with a slight computational element to it because that's what I think is useful for bioinformaticians. So again, I hope you found this lecture useful, and do keep in touch and let me know. And if not, I will see you on Tuesday. Right, take care.